Guys, how are you? Hopefully you're having a better day than I am, because on the day you're watching this for A Day, I'll be I'll be in chemo. Like I said, I hope you're having a better day than I am. Sorry, I'm still fighting this cough, so you'll probably hear me coughing some still as we go through this video. So this video here will work for April the 11th and April the 12th. On let's see, on April the 11th, on Monday, we should have sorry about that. We should have Miss Jarvis in the room. Hi, thank you for helping us out today. And on the 12th, we should have Miss Brown in the room. Hi, thank you for helping us out today. So for both of you, the note sheet that the kids will need is in the I haven't coughed hardly all today. Of course, start coughing right now. Isn't that how it always goes? The note sheets that you will need for all classes on Monday and all classes on Tuesday are back in that doesn't that just figure? Back in that office area on that smaller desk with the wooden top. I think I have it. Man, maybe I should just start all over and do this later. No, we're going to make it work. Onto the stack, I think I have it labeled as April 11 and 12, all classes or something like that. So this video will be about vaccines and immunity. So if you would pass those out to all the kids, and they will have what they need to follow along and fill in the blanks as as I go through these slides. So I think that's all you need for today. So with that said, let me just get rolling on going through these slides. So what I'm going to do when I'm not coughing, I'm just going to go through this at normal speed, and you can pause. You can stop it and pause it whenever you need to if they need a little more time to get all the fill in the blanks. That's what I always do when I'm lecturing. I always wait and make sure everybody has all their fill in the blanks. So you can pause it as you need to. So with that said, give me a second here. Let me switch to the slide view. And we will get started. Get started with these slides. So let me ask you guys, what's the worst disease you can think of? So just go around the room and let them answer and come up with some diseases that they can think of. And then I'm going to move on, but you can pause it as long as you need to. Then the next question becomes, how are these diseases controlled or prevented? So once again, you can just pause it and let them discuss this question. So what we're getting to with trying to prevent some of these things is along the lines of a purpose of a vaccine. So what we're going to talk about today will help answer these questions here about how they're made and how they protect people. And I have two videos on this slide. So let me switch over to these videos and we'll watch these first.
A Surprising as it may seem, vaccination began as a type of traditional therapy at least 1,000 years ago. In India, when a wave of smallpox approached a town, there are tales of people doing something extraordinary. They were lining up to actually buy the disease. The healers, known as Brahmins, would take a cloth and rub the person's upper arm. Then they would scratch the skin, just enough to draw blood. They would then apply dried smallpox scabs taken from patients who had survived the disease. Most people would get sick, but recover. And from that point on, they were protected. Over 1,000 years ago, the Brahmins had observed one of the basic principles of immunity, that you rarely get infected twice.
You got to give people credit. I mean, they got it right. They knew that there was something going on that protected you. They just had no idea what it was or why.
This early form of inoculation was called variolation. Some say it originally came from Africa. Others believe it began as an ancient form of Chinese medicine, where the powder of dried smallpox scabs was blown up the nose.
We know that as early as 1000 AD, the Chinese were using variolation, using smallpox to prevent smallpox. And they were using some very advanced techniques, actually, what we would now call, techniques that you can recognize in modern virology.
Importantly, they always used a weakened form of the disease.
They would sort of select out the mild cases, and they would collect the lymph and put it in a vial and let it dry out and carry it in their pockets for 2 or 3 weeks. So you got at least a semi-killed vaccine. The viruses were, if not dead, certainly, moribund.
From the Brahmins in India, we fast forward around 700 years to Europe, where over 400,000 people were dying from smallpox every year. An English mother, Lady Mary Wortley Montagu, had only just survived smallpox, and she desperately wanted to protect her children. Whilst in Constantinople, she wrote of seeing a practice unheard of in English medicine.
The old woman comes with a nutshell full of the best sort of smallpox and asks what vein you please to have opened. She immediately rips open that you offer to her, which gives you no more pain than a common scratch, and puts into the vein as much matter as can lie upon the head of her needle.
Like the Brahmins, the local women practiced a form of folk medicine handed down through generations.
The children play together all the rest of the day. Then the fever begins to seize them, and they keep their beds 2 days, very seldom 3. And in 8 days' time, they are as well as before their illness.
Lady Mary inoculated her own son, providing him with something remarkable: immunity.
People knew that if you fought a particular disease and you survived it and you were immune for the rest of your life, that was a good thing. So people tried to figure out how can they essentially simulate that infection without really causing suffering and death, but still inducing the immunity that comes from that.
Lady Mary brought the technique back to England, where it was widely accepted. They didn't understand why it worked, and it was never risk-free. But the death rate with wild smallpox was around 30%. With variolation, it dropped to 2%. Fast forward another 70 years to an English doctor called Edward Jenner, who took the next vital step. He proved that deliberate infection with a mild, non-fatal disease called cowpox would protect against smallpox. He called his technique vaccination, from vacca, Latin for cow.
The one thing Jenner discovered was that vaccinia, cowpox, which is generally a very mild disease in humans, cross-reacts, it prevents smallpox. It was actually his only discovery, an important discovery, but people think that he invented the whole idea, which he didn't.
Okay, so I got another one I want to show you over here on herd immunity.
How many people do you know who have had diseases like diphtheria, tetanus, polio, measles, mumps, or smallpox? If you live in the developed world, you probably don't know very many, which is a good thing because those diseases are not only unpleasant but potentially very deadly. And the reason that they are rather rare, if not eradicated entirely in the case of smallpox, is because of vaccines and the power of herd immunity. But what is herd immunity? And how do vaccines figure into it? Let's answer these questions using gummy bears. The very simplest epidemiological model, the SIR model, makes use of three categories: susceptible, the white gummy bears; infected, the red gummy bears; and recovered, the green gummy bears. Susceptible bears can get the disease, infected bears have the disease and can transmit it to susceptible bears, and recovered bears have had the disease so they can't get it again or transmit it to other bears. So here's our model. We have a population of white susceptible gummy bears, and we introduce an infectious disease. It spreads through the population of susceptible bears and infects them, turning them red. Some bears will die of the disease and disappear from the population, but the ones that don't will develop immunity and move into the green, recovered category. The green, recovered gummy bears are important because in addition to perpetuating the gummy bear population, they're able to interact with the red, infected bears without any risk of becoming infected themselves. Assuming that all the gummy bears in our population are going to interact with a certain number of other bears one way or another, it's better for the overall population for red bears to interact with green bears rather than white bears, since the white bears are susceptible and the green bears aren't. As the number of green bears in the population increases, the chances of a red bear meeting a white bear go down until there aren't enough white bears left to sustain the disease in the population. Vaccines are really useful because they allow us to increase the number of green bears in the population without anyone having to be infected first. We can move them straight from the white susceptible category into the green, recovered or immune category. No infection necessary. And if we can vaccinate a large enough proportion of the population, then we can prevent the disease from ever taking hold in the first place, and even the susceptible white bears will be protected from the disease, simply because they will never encounter it. The protection that a high number of vaccinated green bears provides to the small number of susceptible white bears is called herd immunity. Simply put, if enough of your herd is immune to a disease, it's unlikely that you'll get it, even if a few people get sick, simply because infectious individuals are unlikely to encounter susceptible individuals. This is why vaccines are important for public health, and why we don't see many cases of measles, mumps, polio, or smallpox anymore. Thank you for watching this episode of XSciEd. If you have any questions about this episode or suggestions for future episodes, please send us a message.
Okay, so let's get back to our slides here. So, as he was just explaining, herd immunity is the goal of vaccinations for populations. By vaccinating as many people as possible, you can protect the individuals and the population. So that's important. We want to protect not only the population, but the individuals. So if you look at, I got a couple graphs for you here. The infections will drop as immunization increases. So the blue line and blue squares are the percentage of population that's infected. And then the, I don't know, what is that, red or orange? Whatever that other color is the percentage of vaccinated population that's infected. So as we watch the percentage of the infected population and the percentage of the vaccinated population who are infected, they are going to decline as the immunization rate increases. And the rapid drop in infection above immunization rates of 70%, when you get to that point, that's what they start talking about, that it indicates herd immunity. Because you see when you get over here past the 0.7, my pen's not working. There it is. When you get over here past the 0.7, you see these numbers really drop off. So the infection rate in that category above 70% really goes down. So herd immunity also causes a dramatic decline in infection among vaccinated and unvaccinated individuals when vaccination rates are high. So the red or orange, whatever that is the percentage of unvaccinated population that is infected. And then the blue is the percentage of let me try that again, shall we? The blue is the percentage of infected that have been vaccinated. So the percentage of the unvaccinated population who become infected, those are the red or orange triangles, drops after immunization rates are above 50%, indicating herd immunity. And the percentage of those infected who are vaccinated drops above immunization rates of 80%, again showing a herd immunity effect. So that would be about our 50% for those guys, and then our 80% down here. Anything we get over in this range, we see how the infected rate goes way down. And so now we can start talking about again that we have shown herd immunity. So herd immunity prevents the spread of the infectious agent throughout the population, and it protects the individuals from infection. Both good things. Herd immunity can be produced from massive numbers of vaccinations, or it can be produced from massive numbers of infections. Either one will end up producing herd immunity. So a little bit of just reminder, we've had this in slides we did a while ago, but just to reiterate this, the 4 types of classifications of immunity. The first one is naturally acquired active immunity. So this is when you would get sick from some infectious agent, but you recover and now you're immune. So you recover because you have created an immune response to that pathogen, whatever it happened to be. Then there's artificially acquired active immunity. That is the result of a vaccine. And the advantage of a vaccine is you don't have to get sick, but it still produces the same results that naturally acquired does, where we're producing memory cells that keep us from getting sick if we ever encounter that same thing a second time. Then there's artificially acquired passive immunity, where we have antibodies that are injected in a serum from somebody else that had created them, right? But that's short-lived. It's usually created in some other animal. They often use things like horses and things like that are big enough that they can create large amounts of serum. And then there's naturally acquired passive immunity, where we pass antibodies from mother to child during pregnancy or during breastfeeding. Again, these are short-lived, just like when we use the serum. Massive infections can be costly. Wow. Sorry about that. I'm actually not feeling too bad today. Just this cough just won't go away. Anyway, if the infectious agent has a high mortality rate, massive numbers of infections can result in massive numbers of deaths. It would still produce herd immunity once it's spread through the population, but it doesn't protect the individual, and that's a problem. So vaccines are developed for infectious agents with high mortality rates. So they become the target for vaccine development, because we want to produce herd immunity and protect the individual. So for example, have you ever heard of a vaccine for the common cold, which is the rhinovirus? No, because there isn't one. Well, to my knowledge, there isn't one. There's And the reason is because the common cold ends up being no more than just an annoyance. And there's a very low mortality rate for that. So most people will get sick for a few days, but it's typically not a severe sickness. Like I said, it's mostly just an annoyance. So the rationale for trying to develop a vaccine against something like the common cold just isn't there because you don't have the mortality rates. So when they're looking to develop a vaccine, the goal of that is to develop herd immunity. It's to protect the individual by producing both memory T cells and memory B cells. And we're going to talk about that here. Immunity is a result of creating memory cells. So when you encounter some infectious agent, there is both a primary and a secondary immune response that happens. The first thing, the primary response, uses the antibody classes called IgM. They are the B and T cells that get activated for the first time. The B cells become plasma cells when they start making these IgM antibodies. They get activated for the first time. And when we look at this graph here, it's our red line down here. Our IgM is down here. There he is, right? So you notice the first exposure, if we say that's day 0, within the first week, we start seeing an increase there of our IgM antibody. But notice by about 2 weeks, it goes away, comes back down to baseline. Lagging behind that, so right here, the blue line indicates the secondary response. These are where our immune system will switch to have the memory B cells start making antibody and memory T cells, which help them. They will start making antibody, and that category is called IgG. Notice when IgG kicks in, the response is much faster, it's much higher, it's much longer, it's much stronger, all that kind of stuff. And this is what prevents future infection. And notice over here, this is a log scale. This pen just doesn't want to stay on. There it is. This is a log scale. So this increase in IgG antibody in a secondary response, this is a logarithmic increase. But notice for both of these, after day 14-ish, maybe getting into 20 here for the secondary, they flatline again, almost flatline. So the antibodies that they're making are going to not be as prevalent in your blood as they are during this period here when they're actively being made. And what that means is that the antibodies themselves are not what's critical, because the antibodies will go away. The memory cells that make them are what are critical for creating immunity, which also means that if you're being tested, like so many people did for COVID, when they test for the presence of antibodies, the timing of those tests becomes critical. If you time over here, this pen just won't stay on. Come on. There we go. If your timing of your test is over here versus over here, you see you're going to get vastly different results. But the important thing for immunity is what happens over here. Because if we get a second exposure, so you got the infectious agent, you recovered, you're no longer sick, you see the exact same thing again, this IgG response is going to go so high and so quick that it's going to prevent you from getting sick again. That's what immunity is all about. This secondary IgG response that it's so strong, comes on so much faster than the primary response, and you see it can last for, we're out to day 56 here, and we're still really high with our antibody response. It can last for much, much longer than the primary response will last. So that's what's critical for creating immunity. I just said that. Immunity comes from the creation of memory cells. And there has been a lot of confusion over this with COVID-19. I mean, a lot of confusion with this over COVID-19. But no matter what, I'm not going to make any kind of a, I don't know what you say, political or motivated response or whatever. But no matter what has been said, etc., the bottom line is the immune system and the way we respond to things hasn't changed. This is how it works, regardless of what people tell us or what they might be trying to say as far as how the vaccines work, all that kind of thing. This is how the immune system works as far as creating immunity. It doesn't make any difference what anybody might have to say about how it does or doesn't work, but this is how the immune system has always worked. This is how the immune system will always work. And that's the bottom line. So this shows the 6 categories of vaccines that are still in use today. The top row, inactivated viruses, they're killed. So the advantage of that is the vaccine virus cannot cause an infection. He's been killed. The disadvantage is the immune response is not as strong, so you often need more than one dose. So things like polio and hepatitis A and some of the influenza viruses have been of this type. Second one is live, weakened, or what they call attenuated virus. You grow them repeatedly in the lab and they become much weaker, less virulent, as they say. So the advantage of that is there's a strong immune response to this type of vaccine, so you typically only need one dose. But the disadvantage is that there are some side effects due to the fact that even though the virus has been weakened, there'll be still some level of replication of the virus. Examples of this include the measles, mumps, and rubella, chickenpox, smallpox, BCG, which is a tuberculosis vaccine, and some of the influenza viruses, are some of the influenza vaccines that they've developed. Then there is recombinant, where the gene for the surface protein is used in a lab in a carrier DNA molecule called a plasmid to produce a protein that's used for the vaccine. The advantage is that there's no genetic material used in the vaccine, so it can't cause disease. But this is technically more complicated, therefore it ends up being a more expensive form of a vaccine. But this one's been used for hepatitis B, HPV, and again for some of the influenza vaccines. Then we have conjugate, where the protein from the pathogen is attached to a helper protein to cause an immune response, and they call that an adjuvant. Again, the advantage is that we have no genetic material, so it can't cause disease. But the disadvantage is it typically requires multiple doses. Some examples are pneumococcal, the strep vaccines, Haemophilus influenza type B, that's a bacterial infection. The next category is toxoids, where you use a bacterial toxin in the vaccine. Once again, the advantages is we're not using genetic material, so it can't cause disease. but the disadvantages typically requires multiple doses. Diphtheria, pertussis, and tetanus, DPT, that's an example of that. Bordetella, the whooping cough vaccine, that's another example of that. The whooping cough, Corynebacterium, and Clostridium, those are the three examples of the types of bacteria or viruses that are used in the DPT. And last is the new one, the mRNA, which came up with COVID now. They use the mRNA, the M stands for messenger RNA, by the way, for surface protein, and they put that in a micelle, which is just like a little bitty cell, and the vesicle will fuse to the cell membrane to insert it into the cytoplasm of the cell. So the advantage is it can stimulate the immune system to make antibodies and immune killer cells. Disadvantage typically requires multiple doses and can produce side effects and requires cold storage. So the example there for us is COVID-19. And when you talk about the side effects, if you talk to people who've received the vaccine, there's unfortunately a lot of side effects that have gone along with that vaccine as well. So when they make vaccines, they will often use, I use this word, adjuvants. It's an ingredient that helps create a stronger immune response to the vaccine. Typical ones are MF59, which is an oil in water emulsion of something called squalene oil, which is naturally occurring in us and in animals and plants. So it just helps create a stronger immune response. And I was going through this, we saw the flu vaccine showed up in multiple places. There's quadrivalent and trivalent. There's multiple types of those that they're using to that seem to be more effective for different strains. So that's why those show up in different categories. So let me give you, here we got another video. And it's a little bit of history on how they developed the hepatitis B vaccine. So let's find this guy. There he is.
Skin cancer from sun exposure is the #1 cause of cancer deaths in the world. Next is lung cancer, caused mostly by smoking. With both, risk factors are easy to see. But the third leading cause of cancer deaths in the world, liver cancer, is harder to see. That's because it's caused by a virus.
There are about 375 million people in the world who are currently infected with the hepatitis B virus, who are carriers, and they're at higher risk of cancer of the liver and chronic liver disease.
Since his days at Walter Reed, Hilleman had sought a means to conquer hepatitis B virus, but it had remained elusive for one reason. It was virtually impossible to grow in the lab. Then, a young Baruch Blumberg discovered something unusual in the blood of infected people, something the virus was making to escape recognition by our immune systems. Hepatitis B virus made large quantities of one particular protein that sat on the surface of the virus. These little bits of hepatitis B couldn't cause disease, but they could attract antibodies. As many as 500 quadrillion of these proteins would flood the bloodstream, distracting antibodies designed to neutralize the whole virus. If these decoy proteins could distract enough antibodies, then fully formed hepatitis B virus could escape to infect more liver cells. It was a cunning defense. But Hilleman saw these excess viral proteins as the virus's Achilles heel. something he could use to eliminate the disease from the face of the earth.
The use of the Bloomberg knowledge to make a vaccine, of course, you'd say, well, anybody could do that. Well, not really. It took an enormous effort. First of all, recognizing that the source of the vaccine at that time was going to be blood from people who were carriers. They were infected.
Of course, you're handling blood that came from people with hepatitis B infection, which can kill you.
That was not a project that a lot of people were going to be willing to undertake.
It required a whole new concept of how does one make such a vaccine safe.
He had to take human blood. He then had to make sure that any possible virus that could have been in there was killed. He had to take this one protein, hepatitis B surface antigen, and purify it.
He exposed the material that was going to be the end product to 3 steps that would kill any known virus: formaldehyde, pepsin, and urea.
So if you use a thing and it's killed in one step, and the next step it would be killed and killed, it should end up to be deader than dead.
And thus we had the so-called plasma-derived hepatitis B vaccine, a vaccine that was made from, no doubt, the most dangerous starting material that we ever used, that I would argue created the safest vaccine we've ever used.
But just how safe would not be known until the vaccine could be tested in people. Hilleman was at an impasse. Where to test a vaccine that can save millions of lives if it's safe, or possibly give test subjects liver cancer if it's not?
A research scientist probably comes to the hardest point in what he or she will do when human testing is undergone.
You have the responsibility that you might do harm. This is not something for the faint-hearted.
I remember him telling the story about he went into the cafeteria and rolled up his sleeve and got the whole management team in there, and somebody injected him with Hep B vaccine, and he said, now I want you all to do the same. And they were scared.
If you told me you're giving me a vaccine from human blood at that time, obtained from high-risk patients, I'd say, maybe I can do without my hepatitis B vaccine from my good old friend Maurice. No, that's legitimate. That's a legitimate wonder. I'm not saying they didn't purify it enough, but until I see the whole protocol, I'm a little worried. And if I'm a layperson and I don't understand what it means, I'm very worried.
He trusted the science, trusted enough to inoculate himself with this product that started with human blood. So I think, he voted with his arm, if you will.
Human hepatitis B vaccine represents the most technically complex of any vaccine developed to date.
This vaccine gives us, for the first time, an opportunity to prevent this great scourge of mankind.
Maurice Hilleman had become the first person in history to invent a vaccine to prevent human cancer.
Only he had the intelligence and guts and resources to do that. I don't think anybody else would have made that vaccine. It was on the market for 5 years, and it saved lives.
But the use of human blood continued to scare people, and those fears would simmer until they had cause to boil over.
A new disease known as acquired immune deficiency syndrome, AIDS, was first identified about 18 months ago and now has public health officials worried.
Researchers are now studying blood and other samples from the victims, trying to learn what is causing the disease.
AIDS scared people to death. It was clear that certain groups were at higher risk, but because a lot of this was unknown, people would walk into a grocery store and they were scared to pick up a piece of fruit.
There was fear, there was frustration, there was a lot of misinterpretation of how it was transmitted.
Everybody was going to die. 100% lethality.
It is spreading rapidly, cause and treatment unknown. It is a medical puzzle of enormous proportions.
When the AIDS epidemic broke, of course, that really begged the question about using whole blood in a vaccine.
The cause of AIDS wasn't known at the time, but the fear was that perhaps there might be contamination of the vaccine with whatever caused AIDS.
Use of the vaccine waned, and Hilleman was powerless to prove its safety until the cause of AIDS was finally determined by 1984.
Health Secretary Margaret Heckler made the announcement to a jammed news conference.
The probable cause of AIDS has been found.
And she introduced the scientist who led the team, Dr. Robert Gallo.
A test is available for blood banks. The reagents are available to characterize anything now, and a method is available to isolate this virus routinely.
Gallo and others identified HIV, human immunodeficiency virus, as the cause of AIDS. And Hilleman quickly showed that it was incapable of surviving his inactivation process. The vaccine was safe. But still, he found himself up against an enemy he could not defeat with science.
He was dealing with perception more than reality. And the perception was that HIV was sort of hanging over this vaccine like a cloud, even though he had proven clearly that HIV couldn't possibly survive the treatments that he had subjected that vaccine to. So ultimately, he had to abandon it.
It was a tough period. I'm sure it was tough on the company because they invested in making that vaccine. It was tough on Maurice himself. It was tough on Roy Vageras. But again, those were top-notch scientists who managed to come up with the alternative.
The alternative? The new field of genetic engineering. The concept was to harness the manufacturing power of the world's smallest factories, living cells. Organisms like bacteria and yeast are able to share genes by trading rings of DNA called plasmids. These plasmids carry the blueprints that instruct cells to make specific proteins, like the protein Hilleman used to make his hepatitis B vaccine. By using a special enzyme to cut the plasmids, scientists could insert a gene that instructed the cell to make the hepatitis B surface protein. The new vaccine would be easier to make, easier to purify, and just as safe.
The vaccine took time until it was accepted in this country. And then in the year 2000, it was taken to be a global product that is going to be offered to every child born in this world.
It has been suggested that this may have been the single medicine that prevented the most deaths of anything that we have developed in the 20th or 21st century.
The hepatitis B vaccine represented a series of firsts. It was the first vaccine using human blood as a starting material. It was the first vaccine made using recombinant DNA technology. It was the first cancer-preventing vaccine of man. And it was the last vaccine that Maurice Hilleman ever made. It was, in many ways, a culmination of what had been a lifetime of phenomenal achievement of making vaccines, of breaking new ground in many ways with every one of them.
Okay, so there's that one. And back to our slides. So with his hepatitis B vaccine, he used blood from infected individuals as the source of the surface protein, since it couldn't be grown in the lab. But to ensure its safety, he treated it with a series of 3 chemicals to ensure that it didn't contain other infectious agents, formaldehyde, pepsin, and urea. And each one of those on their own would kill every known virus there is. But he took it to the extreme and used 3 times to make sure that the any other infectious agents that might have been in the blood were also killed. So that vaccine was used for 5 years, from 81 to 86. But as the video was saying, when AIDS came out, there was an awful lot of concern about using blood as the source of the material. And that's why they have moved to the recombinant vaccine, which is the one that is still used today. And here's another video on the creation of the mumps vaccine. So here's this one.
Next on Hilleman's hit list, mumps.
Mumps was actually the most common cause of something called aseptic meningitis, a viral infection of the lining of the brain and spinal cord. And it was a common cause of deafness.
Hilleman would spend months trying to find a strain of mumps virus that didn't infect the brain. He found it in the most unlikely of places.
I remember going into my dad's bedroom and waking him up and telling him I didn't feel well. And I think I remember that evening, not so much because I felt bad, but because my dad was quite excited about my feeling bad. It was not a reaction that you expect from a parent.
So what he did was something that no father does. He laid her back down in bed. He went to the laboratory. He got broth. He got a swab. He then came back, woke his daughter up gently, swabbed the inside of her mouth so he could isolate that virus. That was 1963. And there was a mumps vaccine 4 years later. That could never be done today.
The virus strain used in the vaccine is called the Gerald Lynn strain, after Dr. Hilleman's daughter, from whom the original mumps virus was isolated to produce the final vaccine.
Being the cooperative daughter that I am, I came up with the right strain of mumps, and he was able to develop the vaccine.
That photograph of me has haunted me my entire life. People used to say, "Are you Miss Mumps, or was that your sister?"
Gerald Lynn Hilleman, from whom the virus strain was originally isolated, offers sisterly comfort.
Historic moment where an older sibling, rather than bringing the infectious disease home from school, was able to prevent a younger sibling from becoming infected with mumps. So really a game changer.
It really hit me in a very personal way then when my first child was vaccinated, and looking at him and realizing really the enormity of not having to worry about him getting mumps and a lot of other diseases as well. It meant a lot to me as a parent, and it took it from being academically important to really emotionally, very specifically important.
In the U.S. alone, Hilleman's vaccine would cut mumps cases from almost 200,000 per year to just 200, a 99.9% drop. As one journalist noted, Gerald recovered from mumps virus, but mumps virus never recovered from infecting Gerald.
Okay, there's that one. Back to our slides. So the mumps vaccine is a live attenuated vaccine. He found a strain that caused illness but wasn't as severe, and that way the vaccine doesn't have the severe side effects. But the virus also needs to grow well enough in the lab to make the large quantities necessary for commercial production. So researchers often have to test a lot of different isolates of a virus before they find one that's suitable for a vaccine candidate. So he took throat swabs from his infected daughter to get a sample of the mumps virus, and fortunately he found one that proved to be suitable. And it was grown repeatedly in the lab to be weakened, and eventually ended up being the strain of mumps virus used to make the vaccine that is still given to people today, called the Gerald Lynn strain after his daughter. So here's one more video. How scientists make live vaccines.
Human viruses are highly adapted to growing in human cells. These viruses reproduce thousands of times in human cells, gaining access using specific receptors that match molecules on cell surfaces. Like athletes, they get stronger and stronger by continually training under the same conditions. And yet, It's this very strength that makes it possible to weaken the virus. This weakening is the key to making live vaccines. How do scientists make this kind of vaccine? By forcing the virus to grow in different kinds of cells in the lab. In these chick embryo cells, most of the virus particles that were so good at growing in humans can't access the chick cells. But with sheer numbers on their side, a few virus particles are able to unlock the chick cells, infect them, and begin adapting to this new environment. The virus adapts by modifying its genes, but there's a price to pay. As the virus gets better and better at growing in the chick cells, it gets worse and worse at growing in our cells. Now, when reintroduced to the human body as a vaccine, these modified or weakened viruses have a hard time infecting human cells. The virus multiplies less efficiently, giving the immune system the time it needs to gather information about the virus and mount an antibody response. The vaccine provides immunity to natural infection without making us pay the price of natural infection.
Okay, there's that one. Back to our slides. So, why are some vaccinations a one and done and some are required annually? For example, flu vaccines are required every year. So let's talk about that. It all has to do with the type of virus and the genetic material it contains. So there are at least 7 different categories of viruses based on their genetic material. And here are those 7 categories. And I'm going to go through each one of these with you. First is the double-stranded DNA. There are 4 families here that I picked out that are in this category: adenovirus, herpes virus, pox virus, papillomavirus. So colds, flus, diarrheas, conjunctivitis can fall into the category of the human adenovirus. That's one type that causes those things. In the herpes virus family, we have varicella zoster, which is chickenpox, which can later come out as shingles. There's Epstein-Barr, which is mono, and also there's an Epstein-Barr viral disease that can be worse than mono, can last for much longer. There's cytomegalovirus, which also causes mono and some other problems. There's Rosella, which is a rash and fever. And then in pox virus, we have variola, which is smallpox. And we also have human papillomavirus category, which causes cervical cancers and warts. So those are examples of double-stranded DNA. There's partially double-stranded DNA. In here we have HepaDNA viruses, which is hepatitis B. And that's the one we just, that's the one we just saw the vaccine development on, that causes liver infection, cirrhosis, which is damage to the liver, and liver cancers. There is positive single-stranded DNA. The positive refers to that it is the same strand that you would make, that would be the same as your messenger RNA. So what that means is 2 strands of DNA in that molecule, and the one that would be the one that they would make the protein from, that's called the positive strand. In here we have parvoviruses. In humans, there's there's one called B19, which causes something called the fifth disease, which is a mild rash and illness that goes with that. But in dogs and cats, parvovirus can be deadly. That's not something you ever want to hear. Sorry, that's something you never want to hear that your pet has. It attacks the lining of the small intestines, can dehydrate them very rapidly. Then there's double-stranded RNA. Here we have rotaviruses, and these are ones that cause intestinal problems. There's positive strand, single-stranded RNA. Again, the positive means this would be the strand that you would actually make the protein from. You have a number of families listed here. The first one, coronavirus. Man, sorry about that. We're all familiar with this guy as COVID-19, right? And causes all kinds of problems. There's the picornavirus. We have polio, rhino, and hepatitis A. Poliovirus actually is a diarrhea problem in the first place, but when it crosses into the nervous system, it can cause paralysis. Rhinovirus is the category of what everybody refers to as the common cold. And then we have hepatitis A, which causes a more flu-like problem. There's some other, the tonate virus causes a lot of tropical area diseases. Rubella causes German measles. And then there's one that causes hepatitis C. Hold on there. There we go. Then there's positive single-stranded RNA with reverse transcriptase. This is an enzyme that can actually make DNA from RNA. So there's some viruses that have this enzyme with them as well. The most classic of this is the retrovirus, is the HIV, right? The one that causes AIDS, that destroys helper T cells. But there's also the human T lymphocyte virus, which causes T lymphocyte leukemias and lymphomas. There's negative single-stranded RNA. So this would be the one that would be the other strand of the DNA, in this case but it's RNA. But this is the one that would bind to the positive strand. All the bases would be complementary to it and would bind to it. We have a couple families here that I have listed. Influenza, A and B are the orthomyxoviruses. There's the rabies viruses in here. And then we have the paramyxoviruses, the mumps virus, rubeola, which causes measles, and the RSV virus, which is an upper respiratory virus that causes a wheezing cough. It's very common in kids, infants, that kind of thing, but also can come back around and get adults too, but it's most common in kids. All right, so those are our, having a little trouble with the buttons here. Those are our categories of different types of viruses. So I'm going to focus on the question I asked is why sometimes we have to get vaccines every year. I'm going to focus on the flu vaccine. So why do they make a new flu vaccine every year? Well, RNA viruses are the fastest mutating entities on the planet. And if we look at the structure here, they are covered in two types of proteins. One is the hemagglutinin, and the other one is the neuraminidase. The hemagglutinin protein attaches to sialic acid on receptor glycoproteins of a cell, and that's how they gain entry into the cell. So their hemagglutinin proteins are the ones that target the receptors on the cell that help them get inside the cell. Then the neuraminidase, it destroys the sialic acid on those proteins, so new viruses can't infect dying cells, and they have to infect new healthy cells. So it like prevents it from trying to reinfect ones that are already infected. And of that HA and NA, there are different forms. I think they're up to, what I have here is they're up to 16 different forms for the HA and 9 of the NA. And the way you mix those together, they produce different subtypes of the viruses. For example, if you've heard of H1N1, that's what that refers to. The HA would be type 1, and the NA would be type 1. And these things also annually mutate their HA and NA antigens to evade being detected by immune systems. This annual mutation rate is called antigenic drift. They change their antigens. The antigens are the parts that the immune system sees and will respond to. So in this case, since these HA and NA are proteins, they're going to make changes to the amino acids that make up those proteins. So they're trying to stay ahead of detection by our immune system. And then when you have each mutated version of these, it creates a new strain of the subtype. For example, if you have H1N1, we have mutations in there. Now we have H1N1 subtype A, or whatever it happens to be. And immunity is strain specific. Memory cells are created for the specific HA and NA antigens that they originally recognized. So when now we have these new versions and these new subtypes, they will not respond to them. So, new strains annually means that we have to make new vaccines annually against those. So every year, the World Health Organization and the CDC, they pick the strains that are most likely to impact us. And then they make vaccines against those strains. But guess what? This is an educated guess. And it could be wrong, which is why most years the flu vaccines are only 40% effective, because they may not have gotten the most prevalent strain that year. And because, like I said, it's an educated guess based on the ones that they're finding in other parts of the world and the ones that then would be most likely to end up here. Then we also have cases where sometimes the mutations in the influenza are massive, creating versions that can now infect a new species and jumping from things like birds or swine into humans. And this is called antigenic shift. So you may have heard of the swine flu, right? Back in 2009, killed 285,000 in the first year. That was a form of H1N1. Then we can jump back to the Spanish flu in 1918 that killed 50 million. That was another subtype of H1N1. So when we get these new subtypes from antigenic shift, the likelihood is it's something that's never been seen before, and therefore it typically causes a massive impact. So now let me talk to you about the science behind the COVID-19 mRNA vaccines. And this is the dogma of molecular biology, which technically is incomplete. And that's a whole nother story, but we'll leave that for another day. But you'll see this in just about every life science textbook there is, showing you that DNA through the process of transcription makes RNA, and RNA through the process of translation makes proteins. And DNA through the process of replication makes more DNA. So in making these mRNA vaccines, they take advantage of this dogma of molecular biology. So with COVID, the spike protein attaches to the ACE2 receptors, primarily on lung cells. That's the angiotensin converting enzyme 2 in the lungs. So what they did was, to make these mRNA vaccines, they first cloned the gene for that spike protein that attaches to ACE2. And then they inserted the antigenic part of that gene into a plasmid. So again, the antigenic part of it is the part that our immune system would recognize and respond to. And that plasmid is like a carrier DNA molecule that enables you to do manipulation with it in the lab. So once they put it into a plasmid, they utilize a method called in vitro transcription to produce mRNA for the antigenic part of the spike protein from the plasmid. They basically allow it to do what would happen inside of a cell. They allow it to take that mRNA and make protein out of it through the process of translation. In vitro literally means in glass. So it's not done inside a cell. Basically, you add all of the mixture that you would need inside a cell, but you're basically just doing it inside a tube instead of inside a cell. And you add all the enzymes that would be there inside the cell, and you let it do what it does naturally inside a cell, and it will start making the protein that mRNA was designed to make. And in this case, it's the antigenic part of the spike protein. All of this is what people refer to as recombinant vaccine techniques. And by the way, this is something I did in the lab all the time when I was doing research. So then they insert the isolated spike protein into a micelle, which is basically just a small lipid. So it's a mini cell, basically, is what it is. And then they use those particles to inject them into the vaccine. And those particles will fuse with your cell membranes through the process of endocytosis. Because a micelle is basically just a little bitty cell. It has the exact same lipid bilayer that a cell does. So through the process of endocytosis, it will fuse to the cell, and then whatever's on the inside will get dumped inside of the cell. And in this case, it'll be the mRNA. And once it's inside, your cell will start to make the antigenic part of the spike protein from the process of translation. And then your cells will express those antigenic spike proteins on the surface of your cell. Your immune system will then recognize them as foreign and respond with the primary immune response, because it will recognize that as something that's not part of us, since it came originally from COVID, and it will go through the process of mounting a primary immune response. The ones that involve the IgM proteins that we talked about, the IgM antibodies, sorry. I guess I just got ahead of myself, didn't I? That will produce mostly IgM, but it also will produce those memory T and B cells, which will start making a little bit of those IgG antibodies. And once we have created those memory B and C, easy for you to say, once we start making those memory T and B cells, there you go, we now have produced immunity, because they're going to be ready the next time they see COVID to start making their antibodies. And like we said, that response in the secondary response is so much greater than the primary response that will prevent infection and sickness. But remember, there are multiple types of COVID vaccines out there. There's also the DNA vaccines. So with these, they do a combination of recombinant and attenuated techniques, but the DNA vector is inserted, not the purified protein produced from it. So they actually use the DNA to get injected into the host cell and not the protein. So they use the spike protein DNA, which is then made into RNA inside the cell, and that then gets made into protein, the spike protein itself, and that will get expressed on the cell surface, and that will do basically the same thing that the mRNA did, is that it will create the primary immune response and then create the memory T and B cells, which produce our immunity. So 2 different ways they've gone about this, but in essence, it's producing the same type of results. However, COVID has 4 surface proteins. All of this only has involved the spike protein. There's also a hemagglutinin esterase, what they call the HE. There's the membrane protein, which is the M, and the envelope protein, which is the E. So what about these other proteins as vaccine targets? So they chose the spike protein because that's the one that attaches to the receptor to gain entry into the cell. But there's still a lot of research that needs to be done to determine if any of these other proteins are just as, if not more important for immunity as the spike protein is. So we're still at a point where there's a lot of questions that need to be answered for this. And then we've got these issues. COVID-19 is a positive single-stranded RNA virus. So it's going to mutate those surface proteins like all RNA viruses do. But the question is, how frequent? So these are all things that we're in the process of learning. So that means the question becomes, will the current vaccines provide immunity against the new strains? All the other vaccines show us that the answer to that would be no. Once they have changed their proteins and become a new strain or a new subtype, your body will not recognize them as far as the immunity is concerned, and they will see them as something that they're seeing for the first time. And we have to go through the process all over of starting with a primary response again. Does an infection produce the same level of immunity that a vaccine does? And boy, this has been a hot topic that, and you can find people on both sides of this. However, something that I didn't mention is when you're just doing one protein, like they're doing with the spike, and one antigenic region, that's what they call a monoclonal response, meaning it just is focusing on that one spot. People who get an infection and recover from the infection produce something called a polyclonal response, meaning that we have responded to a bunch of different spots on that virus, which probably will include all 4 of those proteins on the surface that I mentioned already, which means a polyclonal response always produces greater immunity than a monoclonal response. So then next question, does everyone need a vaccine? Because you hear a lot of people talking about even if you've already had the infection and recovered, you still need to be vaccinated. But that actually makes no sense scientifically because they have a polyclonal response versus a monoclonal response, which I just explained is going to produce greater immunity. So then there's the issue of should everyone be forced to get a vaccine? And with that, we're getting into, I guess, some politically motivated type of discussion, which I'm going to avoid at this point. But my point in asking these types of questions, you see that there's not always a straightforward, easy answer. And you can go out there and find people on both sides of the fence for questions like these. So with your own health, you're the one that will have to make those types of decisions when it comes to whether or not you want to get these vaccines, that kind of thing. You will be responsible for making those decisions yourself. And I think that is my last slide for today, for this section. So we will end there. And I just thought I'd come back and say goodbye to everybody. The day 2 this week, I'm going to make other videos for you to have slides on those. So goodbye for now, and I will see you on the next video.